we recommend that you use the speaker. There you go. We recommend that you use the speaker rather than gallery view for the presentations. That ensures that you see the person speaking rather than the whole of the room. And if your internet dips out and you have to rejoin, we'll keep an eye on the waiting room and let you back in. And as I say, there'll be time for questions after all 3 presentations. And please feel free to either write any questions you may have and put them in the chat box at any point, or else when we get to the Q&A section, please raise your hand virtually and we'll invite you to ask your questions then. So it's my very great pleasure to introduce our first speaker, Dr. Anthony Khwaja. Anthony is a consultant ophthalmic surgeon at Moorfields Eye Hospital and an associate professor at the UCL Institute of Ophthalmology. Anthony is going to be talking about what the genetic code is and how he aims to build prediction tools based on that genetic makeup that will make treatment of eye diseases more personalized. Anthony, over to you.
Thanks very much. It really is a pleasure to be speaking to you this morning and kicking off this interesting session about genomics and really the genetic code. And what is the genetic code? Well, the DNA, the genetic code in the DNA is also called the code of life. And it's because it is the blueprint for who we are, what we look like, how we behave, and our risk for certain diseases. Eye color is very genetic, and actually we've solved this with really nearly 100% accuracy. If I know each of your genetic codes, I could predict your eye color without ever having looked at your eyes. We're almost there with hair color, another very genetic feature. We can about 90% chance of predicting the correct hair color. But I love this picture of Emma Stone, the actress, as a reminder that it's not just about our DNA, it's how we interact with the environment, in this case, hair color dye, that might actually ultimately influence how we look or behave and so on. But what is the DNA code actually? I love this, it's now no longer there, but this installation that was at the Wellcome Collection Museum on the Euston Road, which has massive double height ceilings and this huge bookcase reaching all the way up to the ceiling with these big volumes on them. And you can see some are open here with tiny print. The print's so small, but when you zoom in, you can see that all of the letters in all of these books were either A, C, G, or T. In fact, this bookcase had 3 billion letters in all the books, all either A, C, G, or T, and that's the genetic code. Now, what's interesting is if you take just 1 point out of those 3 billion, in this case here, the letter C, and if that was a letter A instead, for whatever reason, a mutation, just that one change on its own can cause diseases like cystic fibrosis, sickle cell anemia, some of the other inherited eye conditions you'll be hearing about later from Alison and Jim. And also some forms of breast cancer are caused by just one change in the genetic code. And that was quite famous. As we all know, Angelina Jolie decided to have her breast surgically removed to reduce her risk of cancer when she found out she had one of those mutations, just like her mother. Now, a lot of the more common diseases that affect humans, including the blinding conditions, aren't caused by just one change. They're caused by lots of variations across the whole genetic code, and that's complex disease. Each individual variant on its own doesn't cause disease. So you need to have enough of these contributing factors that together reach a threshold to cause disease. Now, research in this area previously was hampered because it just wasn't affordable to measure this whole bookcase in one individual. But now it has become affordable. The cost of measuring the genetic code has plummeted. It's become so cheap that you could go to your own high street pharmacy and for a very moderate price, actually measure important markers across your whole DNA code. If you wait till Christmas, it's even cheaper. Now, what I'd like to talk about is how I use the genetic code in our research and in my group, so we can start to improve the care of some complex diseases. And I'm particularly interested in glaucoma, which is actually one of the most genetic complex diseases there is in humans overall. Now, there are some challenges when we try to manage glaucoma. One of the challenges is when people have the early stages of glaucoma, they don't even notice it. because these blurry patches go unnoticed. By the time they do develop symptoms, it's too late, because in glaucoma, we can't repair this vision. It's not reversible. So you would have thought, well, we need to detect it early and treat it, right? But population screening isn't currently recommended because our tests aren't good enough when we apply them across the general population. Another challenge is our busy glaucoma clinics. Each dot on this moving graph is an individual patient, and how they're moving is how their vision is worsening over time. Now, if you're a patient, you don't want to end up in this bottom right-hand corner, because that means you've become blind in both eyes despite our best efforts. A major challenge is we can't predict who these patients are going to be in our clinics accurately enough currently. The other, well, it's a problem. It's also very helpful to have so many different treatments for glaucoma, drops, lasers, different types of surgery. But the problem is we don't know how well each individual treatment would work for each individual patient. Another way of looking at this is for laser treatment. If all our patients were the same, they'd have this excellent initial drop in their eye pressure, which is what we're trying to achieve, which slowly wanes over time. But our patients aren't all the same. They're individuals. And these are the individual treatment responses. And you can see some people do excellently well, like super well. Others actually get worse, and a lot do in between. And it would be much better if we could avoid laser in those people who aren't going to do so well with it, and just focus on doing it on the patients that will do really well. So I believe, given glaucoma is so genetic, that the answers to these challenges lie in the genetic code. Now, back around 5 years ago, this graph summarizes our knowledge of genetics in glaucoma. Now, this type of plot is a way of summarizing a study where we look across the whole genetic code and its relationship with glaucoma. Each little dot is one letter in that bookcase I showed you, and how high up the dot is how likely it is that a change in that part of the genetic code contributes to glaucoma risk. And so what you want is the dots to be above this red line, and then you'd say that this part of the genetic code is important for glaucoma. Now, what you want is these skyscrapers, and you can see why these plots are called Manhattan plots. But you could see here, really, it's not looking very much like Manhattan, with only a few buildings reaching the skyline. So the big game changer came from the UK Biobank study, which was a huge study around the country, but really would, from the lobbying efforts of Professor Sepeng Kaur, Paul Foster, from Moorfields and UCL, and with the support of Moorfields Eye Charity, we were able to get eye measurements into the latter part of the study on over 100,000 people, including this test, which is measuring the pressure in the eye. And when we looked at how genetics influences the pressure in the eye, which is the key way people get glaucoma, now look at our Manhattan. This is looking much more like the real Manhattan, lots of skyscrapers. In fact, 133 of them. A real game changer, because what this means is we can now take people's genetic code and give them one number for their genetic risk for glaucoma. And what you can see in this graph here is, according to that genetic risk number, which we can work out at somebody's birth, because the genetic code doesn't change across life, we can work out 10% of the population at 15 times the risk of glaucoma. And what this means, and we're testing this now, is we think if we target our screening test to just these people at high genetic risk, it will work, it will be effective, and we can pick up those people at the highest risk of blindness early to prevent blindness. Now, those studies I've shown you so far have just compared people with glaucoma to people without glaucoma. What we've been lacking is genetic data on people with very detailed information on how their glaucoma has been treated over many years. Did they lose vision? How did they respond to the different treatments? And that's what we're building with the help of Moorfields Eye Charity at Moorfields, the Moorfields Glaucoma Bioresource. We're linking this type of worsening of vision data over time, this type of data about response to treatments with genetic data, and we're building prediction models that, given your genetic code, we can predict risk of blindness and response to different treatments, so we can treat you most appropriately, focusing resources on those who need them most, and saving side effects and costs for people who don't need it. Now, lastly, I just want to end up on something important to me, which is caffeine, and important to many of our patients, but modifiable factors. If we're going to identify people at high genetic risk of glaucoma, we want to empower them with behaviors to reduce that risk. So we looked at caffeine. This graph is a little complicated at first, but we divided up the study into 4 groups according to their genetic risk. We want to find those people at the lowest genetic risk. But if you focus on people at the highest genetic risk, it's really mainly these people, as they consume more caffeine, the higher the pressure in their eye. And so it's only people at the highest genetic risk. And this graph is similar, and it's looking at the proportion of people with glaucoma. And it jumps up at the highest levels of caffeine intake here, but only in the people at the highest genetic risk of glaucoma. So really, the advice we give people, including maybe you should consume less caffeine, is going to be dependent on their genetic code as well. So hopefully this is a glimpse to how at Oriel, we believe we're going to be delivering the next genomics-enabled era of advanced eye care. We're going to enable population screening of those at highest risk so we can pick it up early and prevent blindness. We'll know of the many patients in our clinics who to give the most intensive treatment to and who to spare the side effects from. We'll also be able to pick the correct, best likely to work treatment first off for our patients And I think in the future, we'll also be using a genetic code to give personalised lifestyle advice. So thanks for your attention for this part. It's now my pleasure to introduce Alison Howcastle, who's Deputy Director of Semble Professor of Experimental Ophthalmology at the UCL Institute of Ophthalmology. She'll be talking to us about advances in genomics and patient stem cell models of disease to test potential therapies. Thanks very much, Alison.
Thank you, Anthony. Hopefully everyone can see my slides. Good morning, everyone. My talk is really about the focus that we have in our labs, which is on rarer diseases, unlike the ones that have just been described by Anthony, which are more common diseases. The rarer diseases tend to run in families, and we're really looking for one genetic change rather than a collection of changes of risk factors of disease. Now, individually, these conditions are rare, as I've said, but actually collectively, they are the most common cause of sight impairment in children and young adults in our population. And these untreatable genetic diseases are a real burden with profound impact on the quality of life for these individuals. To give you an idea of the fact that they're rare, but actually collectively common, even if we just look at retinal diseases alone, 1 in 2,000 individuals will suffer from an inherited retinal disease in our population. And although we've identified about 250 genes that cause these inherited retinal diseases, still half of our patients don't have a genetic diagnosis. So we've still got a long way to go to find all the causes of these conditions. And this here is a family tree, just to give you an idea. This is a Moorfields family. And what's really important is to have this information together with the clinical information on the condition that runs in this family. And hopefully you can appreciate this, the filled in circles and squares here are individuals in the pedigree who actually have this blinding condition, whereas the open symbols, they have not inherited the change. So we can use this family tree information together with the clinical information to track genetic changes through the family and hopefully try and identify the genetic change that is causing their condition. And I'm delighted this family has been with us for a long time at Moorfields, and we've just solved their condition. So we're very excited about that. And I'll try and describe now to you how technological advances in our field have really helped us to solve genetic questions, such as the one that we were presented with in this family. Inherited eye diseases aren't just of the retina. So I wanted to just really emphasize that they can affect all cells and tissues in the eye. From the very front of the eye, for example, this condition here, which is a corneal dystrophy affecting the very front layer of the cornea in the eye. This condition affects a slightly lower layer in the cornea, and this individual is just 1 year old, and hopefully you can appreciate these will lead to vision loss and blindness. And here's some examples of different retinal degenerations. There are many, many different types of retinal degeneration. Hopefully when an ophthalmologist looks in someone's eye, they will see a nice, healthy, pinky, orangey tissue, which is the light sensing tissue at the back of the eye. And I'm sure you can appreciate that these are not looking healthy. They are not a nice, even, healthy glow, and the blood vessels don't look very good too. So this is a degenerative disease where we're losing the light sensing cells at the back of the eye. And inherited conditions can affect all tissues from the very front through to the very back of the eye. Now, Anthony's described the human genome to you already, so hopefully you've got some background information here. Here's the human genome spread around all your chromosomes that are in every cell in your body. Now, what's important to realise, even though we found 250 genes that cause retinal degeneration, and we're still missing quite a few, up to half, is that actually genes that code for the proteins only make up about 2% of your DNA that's spread across these chromosomes in the human genome. However, on average, we have about 20 to 25,000 differences in those gene sequences that make up the 2%. And this is what makes us individual, as Anthony's described, such as hair colour. So this is an important normal variation in our human genome. Now, if we look outside of the 2% of the DNA that codes for genes, we actually have 5 million differences in our human genome in these 3 billion letters of our non-coding DNA. So hopefully you can appreciate that we can have differences in our genes, which are only 2% of the DNA that can cause disease, but we can also have differences in the non-coding part of our human genome that can cause disease. And this is something that we're only really beginning to appreciate with the technological advance of being able to sequence the whole human genome, which is now affordable for our patients at Moorfields, as Anthony has just described. So we can now sequence the entire human genome, 3 billion letters for the patients in these families that come to us with these inherited conditions. However, the challenge, of course, is now we have 3 billion letters to read, and it's extremely difficult to analyze this data and determine which of the changes is causing that particular condition in a family. So we need massive computers for all this data, and we need teams of scientists to actually analyze and interpret the data. but it's absolutely transformed our understanding of the human genome and of types of mutations in the human genome that can cause inherited diseases. So rather than finding mutations in the 2% of the DNA in the genes, we can find mutations that do different things now that aren't in the coding regions. They're in non-coding regions that make up the majority of our human genome. We're finding mutations that skip important parts of a gene. We're finding mutations that either delete, insert, or invert big or small pieces of a DNA, so they change the DNA structure. And we're finding mutations, and I'm going to show you an example of something we've recently discovered, of single changes that can switch genes on and off. So it's not actually in the gene, it's in the non-coding region, but it's responsible for determining where and when a gene is switched on and off. And here's an example of one of our recent discoveries. This is a corneal dystrophy, similar to the ones, in fact, it's one of the patients I showed you on one of the initial slides. And we did whole genome sequencing, all 3 billion nucleotides in this family. And we were tracking through the family to look for a single base change in those 3 billion letters that might be associated with their condition. And what we found was this mutation here in a non-coding region. So this line is non-coding region, and this box is a gene, a little bit downstream. So let's say going from King's Cross to Euston Station, that far away. So here we have an unaffected individual who doesn't have the mutation. Now this gene should not be switched on in the cornea, and that's what we find. It is not switched on in the cornea of unaffected individuals. However, we hypothesized that this single change might, in fact, switch the gene on inappropriately in the cornea. So it's a gene that the cornea doesn't want, it doesn't need, and if you switch on, it might cause disease. We were extremely fortunate that one of the patients donated their tissue. They had to have a corneal transplant surgery for their condition because they were blind. And they donated their tissue, and that came straight over from the clinic to our labs. And here, hopefully you can appreciate, if you look at this top line here, this is some corneal tissue from an unaffected individual, and you'll see there's just this pinkish stain, which is normal. In other words, the gene is not on in the cornea. When we look at the patient, hopefully you can appreciate there's this browny orangey staining here in the tissue. That is this gene switched on incorrectly in the tissue of that patient. So we're remarkably grateful to that patient for donating their tissue because this has really confirmed our hypothesis and helps us understand how this kind of changes in the non-coding DNA can actually cause disease, which is a new phenomena that we didn't really understand a few years ago. So human genome sequencing has really revolutionized how we can detect mutations that cause disease. But sometimes it's not obvious how these mutations cause disease. Luckily for cornea, we had patient tissue, but we don't often have access to tissue. So another technological advance that's really revolutionary we can do in the laboratory is our ability to now reprogram skin cells and other cells into what we call stem cells, which we can then change into and make into different types of eye cells in a dish in our laboratory. So I'll just briefly take you through what we do. It's hugely exciting. We can take a little skin biopsy, we call it a badge of honor, from a patient, and we take it straight over to the lab from the clinic, and we put it in a dish in the lab. And here's a picture of a skin sample with the cells growing out of it. So here are the skin cells from a patient. We can then add a little cocktail of molecules that then change these cells into what we call pluripotent stem cells. And here you can see a stem cell colony under the microscope in our lab. Now what we can do for the very, very first time ever is make human eye cells in a dish in the lab from these cells. So now we can make these stem cells into corneal cells. This is some cells we've just grown recently in our lab for a corneal dystrophy patient that we're looking at. So these are corneal cells. We can grow them into neurons, and here's an example of retinal ganglion cells. So these are the neurons in the back of the eye that we can study for optic atrophies and the condition that Anthony's just described, glaucoma. So these cells are really appropriate for studying that disease. And this little beauty at the bottom is what we call a retinal organoid. This is a three-dimensional ball of cells that contains all the cells in the back of the eye, in the light sensing layer called the retina, in 3 dimensions. So this has many different cell types. We can study all the different types of retinal degeneration, retinal diseases in these little beautiful 3-dimensional retinal organoids. Now, why is this important? This is important because we need to understand the genetic condition in this individual. How is it affecting the specific cell types that are dying in disease and causing blindness? So this has absolutely revolutionized what we can do in our research in the laboratory. And here's an example. This is a patient. So this is the question: how are the genetic mutations altering the genes and proteins in the eye? What is the mechanism of disease? We need to understand that if we're going to develop really effective therapies for our patients, which is our ultimate goal. So we'll call these retinal organoids, these 3D-dimensional balls, we'll call them a retina in a dish. And here's a microscopic image of that retina in a dish. In this unaffected, if you hopefully can see this green staining here, this region here is the light sensing cells at the back of the eye that we've grown in the lab from an unaffected individual. Now this gene called RP2 should be expressed in all of these light sensing cells, and you can see we've got green everywhere. That's the normal situation. In the patient from Moorfields, hopefully you can appreciate there is absolutely no green in these retinal organoids. So now for the first time, we can see that that mutation means that this gene is not expressed in the light sensing cells, and it should be expressed. And what's quite remarkable actually is we've been growing these retinal organoids for over 220 days, a long time. They need to be really looked after in the lab. The scientists are amazing. And we can actually start to see this retina degenerate. So it's really recapitulating what happens in human disease. This is a remarkable model system that we can use. Now, not only can we understand mechanisms of disease, but we can then start to do some preclinical development and testing of new therapeutic approaches for these patients. So here again is our patient with no RP2 gene. What we've done here, and Professor Bainbridge will be talking about this after my talk, so give you some more insight into how we can take this into the clinic if it works. What we've done here is we've given the retina in a dish, the gene in a virus. This is called a gene therapy approach or gene supplementation. So we've used this approach for therapy, and hopefully you can see here now we've got lots of green all through this retinal organoid, this retina in a dish, and it's lighting up the light sensing cells like a Christmas tree. So what's fantastic here is we can show that we can introduce the gene back into a human retina, and it will go into the light sensing cells, and it will restore function in those light sensing cells, which is absolutely amazing. So we're going to use, or we're continuing to use, this type of stem cell model to develop lots of different new therapies for these inherited blinding diseases that affect all the layers in the eye. Like I said, from cornea through, we can test drugs, we can test gene therapy or gene supplementation, like I've shown here. One particular approach I'm excited by that we're testing a lot in the lab is something called antisense oligonucleotides, and these are little pieces of DNA or RNA that essentially are like a sticking plaster over the mutation, so that the cell is fooled into thinking there is no mutation there. And this is a really neat new development that we're hoping will really help us take new drugs into the clinic soon. And then, of course, there is gene editing, which I'm sure you've heard about, which won the Nobel Prize, where you can actually genetically manipulate the mutation. So let's say this patient here doesn't have any RP2, maybe we could go in and gene edit the mutation out so that it goes back to a normal gene. So that's the next challenge for us. And I hope you can appreciate how exciting these technological advances are, and our ability now to do this kind of research in the laboratory for preclinical development, and then we can have conversations with Professor James Bainbridge and develop potential trials for any promising drugs or therapies that we see in the lab. Before I finish, I just want to say, hopefully you can appreciate the importance of Oriel in everything that I've described today. Patients are at the very heart of what we do. We're extremely passionate about our research. It's very, very important that we have the connection with our clinical colleagues who are seeing the patients in the clinic. We have connection with our patients. Our patients can see the research that's actually happening in the building. The scientists who are doing all the computational work with all this huge data, we're going to form a tech hub where they can all share their information, and hopefully clinicians and scientists can all come in and work together to find the causes of disease and hopefully help us find some therapies. And of course, patients' samples are really important to everything we do, and their participation in our research has been fantastic. And without the support of Moorfields Eye Charity, none of this would really be possible. So thank you very much for your attention. I look forward to answering questions later, but I will stop sharing my screen here, and I'd like to introduce our next speaker, Professor James Bainbridge. Professor James Bainbridge is a consultant retinal surgeon at Moorfields Eye Hospital and is UCL Professor of Retinal Studies at the Institute of Ophthalmology. Over to you, James.
So I think you need, Alison. I'm just going to share screen if it'll work. Yeah. So I would like to explain how we can begin to use some of the information Alison has so cleverly explained to be directed towards the development of treatments for children and adults with blinding genetic disease. So, as Alison explained, genetic disease of the retina causes severe sight impairment, It's relatively uncommon compared to some other conditions such as macular degeneration, diabetes, but the distinction is that it is very severe and affects people from early life. So it perhaps affects in the order of about 30,000 people in the UK, about 1 in 2,000 people. But because it affects children and affects their quality of life and their ability throughout their life, it has a disproportionately severe impact on their quality of lives, and indeed on the economic financial consequences for themselves and their families. So the impact in the UK has been estimated at something in the order of £500 million is the weight of the financial burden in total for these kind of conditions. And so, although they're relatively uncommon, they are very important. So the technology I want to discuss very briefly is gene therapy, and perhaps in its simplest form, that involves providing a gene to the eye where it's otherwise lacking. And some of the mutations that Anthony and Alison have been discussing result in essentially the lack of an important gene, which results in the lack of an important protein that's critical for the functional survival of cells in the eye. And it can be astounding how the lack of a single gene, perhaps even a single mutation in one of those letters that you've heard about, can result in devastating consequences for sight. And so the approach for the simplest form of gene therapy, that is gene supplementation therapy, is to provide simply the gene that's lacking to the cell for which it's required in the eye. And this schematic just illustrates the way that can be done, so that the functional gene is identified from essentially analysis in the laboratory, so that the correct sequence can be provided. And this sequence is multiplied millions of times and provided to the eye by microsurgery to inject it into the retina where it's needed. And normally a viral vector, so a packaging system is used in order to ensure that the gene gets into the cell efficiently. And so this is a relatively straightforward approach in principle, and it's now possible because of the amazing developments in molecular biology to understand the genetics, genetic engineering to be able to cut and paste genes and to provide them as a treatment, and indeed developments in microsurgery, where we can now use keyhole surgery to inject suspensions of these genes into the retina in a way which is targeted and very carefully controlled. So in fact, at Moorfields and UCL, we have been fortunate to be really at the leading edge of the development of the technology. And in 2000, we were the first in the world to perform gene therapy for people with inherited retinal disease. And the results of those first trials showed in 2007 that this treatment can result in important improvements in aspects of sight. And so this is a feature of the partnership between the Institute of Ophthalmology and the hospital, supported by a wide range of funders, including Moorfields Eye Charity. And so, having shown that this particular approach can result in improvements, a similar improvement for one form of inherited retinal disease, that is a condition called Leber corneal amaurosis, owing to defects in a gene called RPE65. This is one of the treatments that we targeted, and one of the conditions we targeted. A similar approach developed by some of our colleagues in the States, has actually been licensed in the US and indeed in the UK. So we now have gene therapy available in the UK on the NHS for people with one specific form of retinal disease. And this has been transformative, not just for those children and families affected, but indeed for the field, because it shows how this kind of approach can provide a real difference to their lives. So the challenge now is to develop similar treatments for the many other children who are affected by different forms of the condition. And I've just written here some of the different forms of inherited eye disease that we've been working on. These are a range of different conditions, some affecting children more severely, some affecting rods or cones in the retina, but they're all really important. We have, in many instances, identified the potential for benefit, which has enabled us to generate more funding, which has resulted in a quite ambitious commercial enterprise to be able to afford to be able to do all the large-scale clinical trials that are critical to making this available to people. But this is only possible because of the support of the hospital, the university, and particularly to Moorfields Eye Charity for getting things off the ground. So One example I'd just like to point out is that of ARP1, which is the name of one particular condition, otherwise known as Leber congenital amaurosis type 4. And a few years ago, with Moorfields Charity and the critical support of one of the donors who's on the call today, we were able to generate a new treatment for this condition. And we were able to provide that to 4 infants who have this very rare condition. And because they were based outside the UK, when we treated them, we brought them to the UK for treatment at Great Ormond Street just prior to the pandemic, but we've not been able to assess the impact until 2 of them returned for treatment assessment only last week. And we were really astonished, genuinely astonished, to see the impact that this treatment had had on their lives. So, for example, one of them, who was 2 years old when we treated her 3 years ago, is now 5. And at the time, she could see very little. She spent her time staring directly at the sun and would not look at anything else. She would not look at any objects apart from a very bright light, and she would simply follow the light. But extraordinarily now, with the treated eye, she's able to read, which is really remarkable, and a better sort of impact than I could ever have imagined. And we are pretty confident that is a direct result of the treatment, because in her untreated eye, she really can't see at all. There's another child who came to see us last week from treatment prior to the pandemic, who's slightly younger, slightly more difficult to assess, but he was able to navigate running around, or clearly seeing what he's doing, looking at faces. And his family told us that this is very different to his older sibling who has not been treated. So we are cautiously optimistic. I mean, actually, it's all very exciting. We have to be a little bit careful about interpreting the early results, but we are very optimistic that this indicates a really valuable treatment effect, which would not have been possible without the help of our donor and indeed the help of Moorfields, our charity. And going forwards, we're very excited about the partnership that we have with UCL, the hospital, and the charity at Oriel, to enable us to get off the ground with some of these treatments that we can approach with relatively little funding, but can provide proof of principle that enables us to actually seek additional funding to take things through to clinical trial and for benefit in children nationally and worldwide.